thank you everyone for coming today. We're really excited today because we're going to be talking about the CI600 and CI602 root imaging systems that we have here at CID Bioscience. The title of today's presentation is called Hidden Influence, an examination of root system architecture and crop response to nutrition, irrigation, and climate. And so today we have two guest speakers with us. We're really excited about our guest speakers. First, I will go over some housekeeping, and then we'll go into some introductory material, talk about the actual instrument itself, and then we'll go into our guest speakers' presentations, and we'll end with a Q&A with both myself and the guest speakers. So right off the bat, we do have a fourth person on this Zoom call with us, and that is Scott Trimble. He's our webinar moderator, and Scott is our Director of Marketing and Communications here at CID Bioscience. And he's been with the company now for 3 years. So Scott will be monitoring the chat. He'll also be posting relevant links as we go through the presentation. So whenever you see a post from CID Bioscience in the chat, that is Scott sending you an extra information or supplementary information. Some housekeeping for all questions that you have. If you have any sort of questions relating to the content of the webinar in any way, whether it be about our instruments or about the research that's being presented, please use the Q&A function in Zoom. So there is an actual Q&A function. There's a box that says Q&A, and you can actually just click that and post your questions in there. And then it lists all the questions in order for me. It makes it really easy for ourselves to answer all those questions and make sure that all the questions are addressed by the end of the webinar. If you're going to use the chat function, please keep it to only things of like technical difficulties or just, you know, communications between the participants saying hello or other cordial greetings or things like that. You can go ahead and use the chat function for that. But if it's a question, do not use the chat function. Please use the Q&A function. And it says Susie here, who is usually our moderator, but Scott will actually be the one posting all the relevant links and extra information needed. That information will be in the chat. A little bit about myself, if you aren't familiar with me, my name is Galen George. I'm a application scientist here at CID Bioscience. My bachelor's is in biochemistry and my master's degree is in food science from Michigan State University. I'm an IFT certified food scientist, and my previous experience is in managing analytical food testing laboratories and working as a food safety consultant for the food manufacturing, produce, and cannabis industries. So today, we're going to go over some introductions, get into the CI600 and the 602, talk about what they are, their features, their specifications, talk a little bit about applications, who's using it, how it's being utilized. Then we're going to jump right into our guest speaker presentations. So we have Dr. Rao from Agriculture Victoria and Dr. Lotze from Stellenbosch University. And then after that, we will go into our Q&A, which will be with both myself and our guest speaker. So you can ask specific questions regarding the research. You can also ask specific questions about the instrument, and I can help answer those. And now also they can help share their experiences. So first, a company overview. If you're new to our webinars here, we've been doing these for a while, but if you're new, our company is called CID Bioscience, and we've been around since about 1989. And so we apply more than 30 years of our experience in plant research and instrumentation to help serve the research community and commercial needs. And so what we do is we create non-destructive measurement devices. And these devices help researchers acquire consistent, high quality, and transparent data. We're really proud of the fact that our instruments provide instant, accurate results. They're very durable, they're field ready, they're portable. And we do all of the engineering, testing, manufacturing, design, all of that in our headquarters here in Camas, Washington, where I am at currently. So a little bit of overview about the CI600 and the CI602. What these instruments are used for is essentially getting an entire view of what's happening below ground without having to damage the roots. So instead of having to classically dig up roots and examine them and measure them, and thereby destroying or harming your plant that you're researching. The CI600 and 602 root imagers actually allow you to get a view of what's happening with the root dynamics underground without having to do any sort of damage to the plant. And so since there's a lot of change in the root dynamics seasonally and in response to biotic and abiotic stressors, it's actually becoming a very popular thing to study as far as looking at a holistic view of plant health. And so people are using these across multiple different fields, from agronomy to just plant physiology, environmental science, forestry. And so what these non-destructive root images to show is a variety of features like the root system architecture. You can actually see over time new root growth, dormancy, or root dieback. Look at things like mycorrhizal root tips, potentially parasites, nematode cysts, other kinds of infections. And it's a really easy system to use because all you do is install clear plastic tubes at an angle in the field application, wherever you are, either in a cornfield or a sugarcane field or even in the tundra. And you can install those tubes, leave them in the ground, and you can track the root systems over time. And you can look at them in response to fertilizer applications or watering or drought conditions, all sorts of things. So it's a really kind of all-encompassing tool that does give the researcher a more holistic view. If they're already studying what's happening above ground, you can now get a view of what's happening below ground, and then you get that holistic approach to your research. Some features of the instrument. So you do get very high resolution images. They can go up to 23.5 million pixels. It is a linear scanning system. So essentially how it works is the scan head will rotate 360 degrees and you'll get a rectangular image of your root system, but it's actually the roots that are all surrounding the tube. You can change the resolution to alter the amount of time as well as the resolution of the image. So that goes from anywhere from 100 to 1200 dpi. For the 602, the 600, that only goes up to 600 dpi. But as you increase the resolution, your scan time is going to increase as well. So there is a kind of a cost benefit analysis that you have to perform yourself to figure out what is the resolution that you need for your application and how much time do you want to spend taking your scans. It does, so as I mentioned, it does take a 360-degree scan of anything that's within a few millimeters of the tube. So as long as the root is touching or within a couple millimeters of that tube, then it will be imaged. But if it's further away from the tube than that, then the light is not able to penetrate through that much soil, so you actually won't be able to see those. So it is important to be strategic about where you place your tubes when you go to install them in your system. This system also includes a tablet computer that powers the scanner. It operates the control software, so the actual data acquisition software controls that. It also saves the images. And that tablet also is a touchscreen tablet, so you can easily maneuver with that. And it also uses Windows operating system. It is a portable and convenient instrument to travel around with. So if you need to go to multiple remote field locations, you can bring it with you. And as I mentioned, there are two instruments, a 600 and a 602. The only real difference, besides the fact that the 602 can go up to 1200 dpi, is that the diameters are different. So the CI600 has a 2.5-inch diameter, and the CI602 has a 1.8-inch diameter, which means it fits the 2-inch inner diameter tubes. So if you do have tubes already installed from a different manufacturer than ourselves, then it will fit inside those 2-inch diameter tubes. And then as far as analyzing the actual roots goes, we did just talk about the instrument that acquires the image, but how are you going to assess? So the output of the device is actually just an image file. So you can use any software that you want to assess the images. We do offer our own proprietary software called Rootsnap. And so what Rootsnap, and it comes free with the instrument. And so if you want to use Rootsnap, it also is kind of optimized for the touch screen. It's a multi-touch interface, and it provides measurements of root length, area, volume, diameter, and branching angle. and it can report the average values along with individual root values. It also, you can stitch together multiple images to create a larger image. You can also do studies over time where you basically layer the same image over top one another, and you can see root growth and root dieback. And so it does have a couple of other functionalities like snap to root, which helps with the root tracing. It does still require manual tracing. There is no AI built into this software. But it is still an intuitive and efficient software to use if you would like to use it. And it is free, as I mentioned, with the instrument, so you don't have to go and try to purchase a license for a different software unless you so choose. Some applications I've already kind of talked about in that first slide. I already talked about some things that the CI 600 and 602 can be used for. Mainly, the number one application is just observing the function of a plant's root system over time. So whether that be in an agricultural setting, as I mentioned, or in a more environmental science or forestry or other kind of setting like that, that's the main function of the instrument. People do use it to track establishment and rooting of new crops. As they plant, they actually install the tubes before they plant, and then they can observe how the crops are established. Also, we have been seeing more people using it, as I mentioned, for a holistic approach to looking at early disease detection. So especially Dr. Rossi down at University of Florida, who we've spoken to about before about the 600 and the 602. He uses them for looking at helping to get a holistic approach to early detection of HLB or citrus greening disease in the citrus crop down in Florida. And so that is another application that we've been seeing this used for. And then other things like timing soil amendments, applications looking at root flush, investigating efficacy of fertilizers, and other sorts of treatments, and looking at efficacy of treatments is another big use for the 600 and 602. But you don't have to take it just from me. We actually have two guests on the presentation today who will talk about their specific applications and how they're utilizing the root imaging systems from us. And our first guest is Dr. Elmi Lotze, and she is a senior lecturer and researcher at Stellenbosch University in the Department of Horticultural Science. And that's in South Africa, for those of you who are wondering. So she's, it's late night over there. And she attained her PhD in 2005 and has held various positions in the fruit industry. And so she joined Stellenbosch in 2015, and now she is a person who trains post-grad students in horticultural science and sustainable agriculture, as well as teaching applied plant physiology to final year undergrad students, so the more advanced undergrad students. And her current research focus is plant nutrition, climate change, and root dynamics across crops. So she's been doing a lot of studies on redbush tea, blueberries, calcium applications in apple, wind damage in citrus, cover crops in perennial orchards. So really looking at a wide variety of applications here. So we're really excited to hear more from Dr. Lotze, and I will go ahead and stop sharing my screen now. And then once I stop, you'll be able to share your screen, Dr. Lotze.
Karen, can you see it?
Yep, perfect.
Okay, so I will start. Okay, thank you very much for this opportunity to share some of my results. In my presentation, I would focus on a broader idea and less about results, but give you maybe an overall idea of how robust the specific CID 600 is, what we can do with it, and what the potential is. So I'd like to concentrate on the opportunities, results, and then the challenges. And when I started, I was a bit younger. It was a couple of years ago. And I started with my first student working on apples. So we've got opportunities in South Africa, but it comes with a challenge, and that's topography and poor soils. The Western Cape is notorious for bad soils. and we have lots of soils with stones and sand and also clay. So it's quite a challenge if we start to look at the images. Initially, I worked predominantly in apples, and this shows you some of the physical aspects of looking at an apple tree root system. On the left-hand side, I always use a destructive study to get an idea of the two-dimensional idea of what the roots look like. And this is a distribution of a 1 meter by 1 meter pit that we dug to show the distribution of a bearing apple tree. On the right-hand side at the top, you will see quite often if there's a mulch in the orchard, you see an accumulation of roots at the top. That is also interesting when you look at the CID scanning images. And then at the bottom, it gives you an idea of what the soil looks like quite often, and the trouble we have to put these tubes into the soil. An auger, we usually start with an auger because it helps you to be able to look at the images quicker because it stabilizes the soil. But more often than not, we have to also use a spade. So I've got a couple of case studies that had different aims that I'd like to introduce you to show you how we utilize the CID and the images of the CID in different levels. We started off with the easiest one, and that was to look at white root tips and determine the seasonal dynamics. Here you can see white roots, and what we particularly looked at is the white tip. So that was really easy. It was easy to count them because it was only a number that we had to look at. And the main idea was we wanted to correlate that to soil applications of calcium. So what we looked at different apple cultivars, and we quantified the effect of soil, climate, rootstocks, and cultivar on the dynamics of the white root tips. And then we also looked at whether there was a difference between older trees and non-berry trees. Thereafter, I got a bit more enthusiastic, and we started to look at the other features of the software, and we also started to include the number, the volume, and the length of different roots. And in this regard, I tested that or evaluated that with a couple of trials. The one was biostimulants at establishment, and in this case, we also included a study on citrus. I also looked at drip versus micro-irrigation. Currently, I'm looking at the effect of yield and irrigation volume on root development, and then also currently looking at rootstock resilience to drought, where we also used the CID to quantify the root reaction. Typically, what's done is you have to install the tube perpendicular to a tree row, in this case, normally at a 45-degree angle, and then we can cover an area of about between 60 and 70 centimeters soil with this installation. And again, at the bottom, you can see the type of soils that we have to battle with in the Western Cape, if we refer to apple growing. This was some of the first images that we collected, and this shows you the white root growth of a single root, or actually a few roots, but we focused on single roots. And this is a heavy soil. I saw there was a question about a heavy soil in the chat box before, and this was during winter. And here you can see this root starts to develop, it branches, it ages, and then eventually in the last slide, it either died or it aged, and sometimes you lose the initial image just because the root moves. And in this case, it was quite interesting because it showed a lot of root activity during winter that was unknown before. So the first trial was then on bearing golden deletions trees on a specific rootstock, where we wanted to quantify the number of white root tips, and we wanted to see what happens during the season. And also because we know that there's a bimodal growth cycle in the northern hemisphere. And if we look at this graph here, you could see again there was a bimodal pattern that we followed for 2 seasons. So that confirmed the bimodal growth pattern of bearing apples in South Africa. And this was true across cultivars and rootstocks and soil types. And eventually we also confirmed that in 2 different regions in terms of chilling requirement in South Africa. Where it differed from the Northern Hemisphere was with regard to the main peak. And in our case, the main peak was actually in winter. That was unusual because in the Northern Hemisphere, usually the main peak is during the summertime. What you'll also see here is that in terms of the actual numbers, the numbers varied during the season, and that was due to different circumstances. In our case, there was a severe pruning at the end of 2013, and that resulted in a huge number of, or a higher number of white root tips the following season. So that was also something that was very interesting to quantify. At the bottom other graph, again, we looked at the average number of white root tips. And here what I wanted to show you is, in this case, we followed the trends during 4 years. And again, the trends didn't vary, but the number actually varied. And that was related to the above ground dynamics. And that also allowed us to explain the root growth in a dynamic way. And we could correlate that to yield in this case. Then we moved to young trees in an area close by, and we wanted to confirm that the same growth pattern was followed in young trees. But in this case, it was not exactly the same. We saw a more continuous root growth because there's an absence of fruit, so that sink wasn't there. And we also find, in this case, in the graph, it shows a little bit more than 1 season, and you can see it's still bimodal, but the growth continued a bit longer, and it looked like the summer peak was a bit higher than the winter peak. What we also saw here was the effect of the root extension and the importance of the positioning of the tube, especially during the second season, because all of a sudden we didn't find any roots. And then when we dug, of course there were roots, but they were on the other side of the tube. So in this case, we had to remember that if we want to install the tube, and it is in the beginning of this of the life cycle of a tree, it was important to also know that we should have had another tube further along the line, or just remember that there's growth on the other side of the tube as well. What we could also quantify using this technology was that the poor plant performance was due to saturated soils, where we didn't see as much root development as we wanted to, and then we confirmed that when they also discovered that there was a problem with irrigation. So the next trial, we wanted to see if the equipment could also distinguish between different treatments, and we used biostimulants, and we evaluated the performance on newly planted apple trees in fumigated soil. So in this case, again, the soil was actually easier to install the tubes. The trees were planted, the treatments were applied, and then we looked at results. So this is only one set of results. What I wanted to illustrate here is that in this case, we looked at the total number of roots, and we looked at the different levels. So in this case, soil level 2, that is close to the bottom, 30 to 45 centimeters. And in this case, we could use the equipment to distinguish significantly between results of the treatment. So in the 17th of January 2017, it was possible to show differences in root reaction based on the treatments that we applied. So that was quite neat, because in this specific trial, we didn't see a lot of results above ground. So it was really, really nice for the student to at least show some significant differences in the soil. Then we moved to Ceres. It's another area in the Western Cape with much higher chilling. And here we investigated the tree performance of young non-bearing planted trees on M9, although in the 3rd year it started bearing, so we started to get a result. The main thing here was it was irrigated by microjets and drip irrigation. And in this case, it was a narrow planting in much younger trees, of course, and it was 3 meters by 0.8 meters and planted on a ridge. That presented us with another challenge, because now the tube had to fit onto the ridge. So in this case, we installed the tubes about 10 centimeters from the trunk, whereas previously we used about 40 centimeters away from the trunk to get some kind of a root distribution. On the left-hand side, you can see again a pit that shows you the initial development of the roots. That was quite shallow, and then it started to develop thereafter. So if this is some provisional results after 3 seasons, what was different in this case is that the drip was actually applied as a full surface management. So the drip and the micros both covered the whole surface area. After 3 seasons, we saw significantly bigger trees and a higher accumulated yield in the drip. And also we looked at the destructive root analysis that indicated more roots in the top 70 centimeters for the drip than the micro in the 3rd season. And this, we also saw a different water use pattern in the drip versus the micro. So what, in this case, we looked at the average root length, again, for the whole tube, for the whole 60 centimeter tube. And here we could see changes in the 2nd season only. So in the 1st season, the student wasn't that laborious. And initially the micro had less roots than the drip. In most of the cases, there's an exception. But in the 2nd season, we started to see some significant differences at specific times, where again, the micro had less roots than the drip, and we still need to follow these results up. What was important is that in this case, we still also had to look at the role of fertilization and irrigation volumes before we could describe or ascribe the results only to the different irrigation methods. Moving on to citrus, we looked at the root response of Nadorkut monodrons to biostimulant application, different biostimulants, but in this case, we also wanted to see how the roots reacted. In this case, we looked at the seasonal growth dynamics. So again, now we start to also look at how the whole root performs and not only the white root tip. Here you can see what the roots look like. Typically what happens if you start too early, you find that moisture can accumulate against the tube. And once the tube is stabilized or is well established in the soil, then you don't see this problem. Some of the root response results, what was interesting is that the treatment reaction was compared between the above and the below soil parameters. So in the graph, you could see that here we show the mean root length as well as the mean shoot length over, between the different treatments at the end of the season. And we could see the one thing that was nice is that we could see a difference between the different treatments, and they differed significantly. But what was also very nice to show is that the biostimulants had different modes of action. And here you can see that these ones stimulated root growth. And then you could see that the root growth and the shoot growth opposed one another. And that's typically what we find in literature. And this was particularly obvious when we looked at the citrus roots. So that was really nice that the root data quantified the hidden treatment reaction that we would never have seen if we didn't have the equipment. So 2 things that was nice here. We could again illustrate significant differences between treatments, but we could also say that when we didn't find a reaction of the treatment above the soil, it was important to also monitor something below the soil. This was a little bit out of my field, but it was a student, a PhD student, that wanted to look at the morphology of the roots in a rain-fed maize trial. This was done upper country, and the soils were lovely. It was easy to install the tubes. It was very nice soil, and there again you see an image of how much different the roots look like in the maize. He focused on different parameters, looking at lateral root length as well as the branching angle at different phases. And in this case, we're only going to present data of one specific phase, the tasseling phase. And he found very nice significant differences between treatments. The one was, it pertained population density. And then he also looked at the differences against the crop rows and also between crop rows. So there were 2 treatments there as well. And he found significant differences at certain depths, 15 to 30 and 30 to 45 centimeters. And unfortunately, 1 year was also during a major drought that struck South Africa. But in this case, you could also find differences between seasons. That was also very nice to show, what the roots do when you find it in a rain-fed maize population. This paper was also published, and again, just want to show you, in this case, it was a lateral root length, but when the tube placement was either against crop rows or between crop rows, we had the 2 seasons, and then we had the 4 different depths that you could cover using about a meter, 1 meter, 10 centimeters, normally what we use for tube length, and it was possible to do 4 scans. And then you could show significant differences depending on what you wanted to look at. The current projects I'm busy with, we just started with a project with red bush tea. In this case, it's a bush, it's a local indigenous bush that you make tea from. What is a bit more challenging is, in this case, there's only like 30 centimeters between the bushes. In this case, I will use a tube that will cover both plants. And we will have to look at interpret the root information for 2 plants, but down to the depth of 60 centimeters. The soil is very sandy, so in this case, we won't be able to use an auger because everything will just collapse again. So although it's easy to install the tubes, in this case, we'll still have to dig because of the sand. It's the first time I'm going to work with blueberries, and in this case, it's completely different because the blueberry has a much shallower system, and we will only use a tube or 1 image per tube of about 20 centimeters depth. And we want to determine the seasonal root growth pattern because it hasn't been done before, and we want to relate that to phenology as well as the carbohydrate allocation in blueberries. Then we're finishing 2 projects. The one is in the north. We will also want to look at the seasonal growth pattern because in this case we want to do that on 2 stone fruit cultivars, 3 peach cultivars actually, and it's to determine whether it differs that much from what we see in pome fruit, and also the fact that we have a different climate on that side, and that's going to be a summer rainfall area. And then the other one that we're finishing off is, again, looking at the seasonal growth pattern and carbohydrate allocation, but in this case, again, on apple. And we want to look at the interaction between irrigation volumes and then also the crop load. So in the provisional results from blueberries from last year showed that here we looked at the total root length of the 2 cultivars. The 2 cultivars have different yields. And the problem is that the one cultivar has a much lower yield than the other, and we want to try and determine why. So there's my student sitting with a small tree, trees or bushes at this stage, and we found that there's no big difference between the total root length of the 2 cultivars, but we found quite a severe difference between the root numbers. So in this case, we will follow this up. What is interesting is, again, looking at a completely different picture when you look at the image of the roots. And this is when I become really, really thankful that I have a student that wants to count the roots. This is an image or a picture of the stone fruit orchard in the northern side of the country. Again, looking at roots and being very fortunate in having a student that is prepared to count the roots. And in this case, you can see it becomes increasingly more difficult. It's not always easy to identify the individual roots. And there's always the question of whether the roots that you see are actually the roots of the specific crop or whether it's root invasion from weeds. Provisional results here. Again, looking at the total root, in this case, only level 1, that's down at the bottom. So the one thing that we could see is that the roots go down to the bottom to that exceed 60 centimeters. These are 2 seasons, season 1 and then season 2. And again, what we see is a kind of a peak here and then another peak there. So it looks like it's also going to be a bimodal root flush. in the different in all the different peach cultivars. Here we see we have two sites, site 1 and 2, but you see the same kind of pattern at both sites. So it looks like in the two different microclimates, we're going to have the same behavior of the roots. And it's also similar for the cultivars. But again, what we see is the number, the actual, if you think about, there's a difference in the root length. So it's probably also going to be in the number of roots that shows you, although the dynamics are the same, you'd find differences. And now we have to quantify whether these differences are cultivar related or is it management practices. Then look at the challenges specifically locally with us. The first one would be the soil type and the root identification. You get heavy soils, you get sandy soils. It's not always so evident to see the roots, although we would like to do that. With a lot of other imaging packages, you can change the color or you can increase the contrast between what you want to see in the background. In our case, it's not so easy, because a root is not white or pink or yellow. The root starts with white and it's got 50 shades of white, and eventually it becomes brown. So it becomes difficult to see it, and that's where the human eye is still important. The installation of the tubes in the orchards, unfortunately, that's how I work. So there are definitely other colleagues that have an easier way to install the roots, the tubes in the root section. With us, quite often you have to bend down, and it's difficult to install it. And then also we have the soil fracture, and that makes it just a bit difficult to install it. But that's just the practical thing. What we also found is sometimes you find poor irrigation management, and this would of course influence the development. And you must take care when you interpret the results. That you are sure that it's what the roots are doing, and the root reaction, what you see, is not due to some kind of management practice that's not perfect. General challenges in using the equipment. The one is the identification of invasive roots into the crop area, and you have to go and dig and make sure if you see different roots whether it can be due to root invasion. Because it's not always so evident to identify the roots and see, well, this root is completely different, or it's got a different shape. Quite often you do find that weed roots have less branching. Then resolution versus time and the size of file. Galen alluded to this. You can increase the resolution, and especially if you want to do more work that's more focused on utilizing the technology to describe, say, root reaction on pests and diseases or more micro work, it would be better to increase the resolution, because then the identification of the roots is much easier, but it takes more time and the file size becomes problematic. The time is manual tracking. Yeah, that again, Galen referred to that. At the moment, it's still a manual thing. You have to use a mouse or a finger, and that's a stumbling block for some students. Selection of the ideal placing of the tube in the field. I indicated this a little bit before. Sometimes it's about having the ideal placing of the tube, but then also, because I work a lot in field trials, that may be in the tractor row, and then you can't do it. So sometimes it's a compromise between protecting the tube, not being damaged, and placing it where you want to find the root reaction, and you must know your crop to know this. And also, people are now increasingly planting on ridges. That makes it a bit more challenging, because the ridge can be anything from about 60 centimeters to a meter, and if it's like 60 centimeters, you really only have like about 10 centimeters to play with. The root growth dynamics, that's a lesson that I learned in the beginning, and that is initially, if a apple starts growing, it grows quite vigorously into the work row, but then it extends another year before it starts to branch a lot. So again, depending on the crop, you may find that placing of the tube is not ideal for the whole time that you want to evaluate the root growth, but that you maybe have to either install another tube or have two trials where you have two different positions of the trial, of the tube. In terms of interpretation, there's always a payoff between statistics and logistics. You can only count so many images at a time, and then you become a bit aggressive or disheartened. So you can only do so many images in a lot of detail, but then also there's the statistical side that needs attention. We also have to accommodate the plant to plant variation. It's always there. We're working with living organs, and you do find that some plants just that they have just a different root reaction than another one. Sorry. And therefore you can't only rely on one or two tubes. I normally take at least 3 tubes per treatment, so that if there's a plant-to-plant variation, at least you can still get some kind of feeling of what the average would look like. In terms of another thing, in terms of interpretation, is how to relate what you see in the Mycorrhizal to the actual root volume, and that's why I always prefer to also do destructive root analysis to put it into context. And then finally, there's a lot of potential to integrate the Mycorrhizal images with physiological and phenological data, and that's where I really find it very beneficial. Adaptation since 2013, when I started using the equipment, an improvement of the power use of the scanner. I used to have to go and drink some tea so that the battery could charge, and now it's not necessary anymore. The availability of the tablet with the touchscreen makes a huge difference, and it's easier to also to work with that in a field situation for rough trials. And of course, then the online assistance is improved as well, and our access to research results in this field. So acknowledgement, I did this with a lot of students and also some co-supervisors, and of course we have funding for most of the trials. And then I've got some publications for people that are interested in more data on using this. So with that, Galen, I'm done.
Wow, thank you so much, Dr. Lotze, everyone. That was very in-depth and very interesting to hear about all the applications you've used that for. So let me go ahead and reshare my presentation. And I know I already saw we definitely have some questions that'll be specifically geared towards you at the end of this webinar here. So next, we have another guest speaker, and this is Dr. Shiwangni Rao. And Dr. Rao is a research scientist at the Agriculture Victoria Research Department of Plant Molecular Breeding. And so her recent work is in pulses, pre-breeding involving looking at pulse root system architecture for trait selection and overcoming production constraints. So she has a master's from the University of South Pacific in Fiji and her PhD from Charles Sturt University in Australia. And so obviously with this a focus of looking at pulse root system architecture, overcoming production constraints, she has a background in environmental science, and she's focused on looking at food security and food supply, which is something that I also am very interested in. So Dr. Rao, I'm actually going to stop sharing and then I'll let you go ahead and share your screen.
Hi, can you hear me?
Yep, we can hear you just fine.
Okay. All right, I'll just... Okay, awesome. Yes, thank you, Galen, for the introduction, and I'll try and be as quick as I can because we seem to be a bit short on time. So yes, I'm Shuang Ni Rao, and I am doing some root system architectural study of lentil. Actually, there's quite a few pulses, but the focus for today's talk will just be on lentils. Very quickly, my project looks at the physiological and agronomic practices that we can use to overcome pulse production constraints in Victoria. And the project is funded by GRDC, which is a farmer-funded organization, with Agriculture Victoria, which is the state government's agricultural research sector. And we are in our second year of the project. And so using the root imager, we just started last year. Last year was our first field season, and it was a preliminary trial. So I have a little bit of background and a little bit of coverage on the CI 600. So the background, my project basically is in 3 modules. The first one was to identify the constraints that were present in the growing region. And from this, we've done a desktop study and produced a publication, which is on the pulse root idiotype. The second part was, once we've identified all the constraints and the growing regions, what traits would be most applicable, or what traits would form the best idiotype for the best production. But unfortunately, there has been no screening studies done on lentils or peas from the Australian perspective. And this is quite important because the Australian climate is a bit different from all its counterparts. And so these varieties that are released in Australia are quite specific to that particular growing region. And then once we have identified the diversity of root traits and shoot traits that are present in our germplasm, to then find out what are the best management practices, coupling it with agronomic practices to get the best yields, and also doing some crop modeling to see what impact different conditions have on this idiotype and its production. And today's talk comes from the second module, which was to look for genetic variability in terms of root traits in pulses. So a bit more on the introduction. Why pulses? Basically because pulses are a superfood. They have pretty much everything you need in your diet. It's got protein, fiber, foliate, iron, calcium. It's low in fat, and it's not only good for human food, but as well as stock feed. And apart from that, it's great for the agricultural system because pulses fix nitrogen into the soil. So cereal crop after a pulse crop benefits a lot from this. Also because pulse roots seem to have a bigger diameter than cereal crops. It leaves a lot of biopores in the soil, and studies have shown that when you have a pulse cereal rotation, the cereal crop ends up having a lot more root growth due to the biopores that pre-exist from the pulse crop. So yes, they are an excellent crop to study. And in Victoria, in Australia, pulses are mostly grown within these regions. And though they look very small on a scale of, say, to the country, they do have very diverse microclimates happening within the region. And you can divide these into low, medium, and high rainfall areas. In the low rainfall area, the rainfall's around less than 350 mm. The soil is very sandy and calcareous, and drought is the main constraint. In the medium rainfall zone, we've got 350 to 550 millimeters of rain. The soils are calcareous soil, soils, and vertisols. Once again, in reference to what Dr. Elmi was talking about earlier, these and the question on heavy clays. Vertisols are very heavy clays. They are also known as cracking clay because when there is rainfall, it absorbs a lot of moisture, and then during summer it dries out and leaves huge cracks in the ground. Like if you drop your phone, you may not find it, kind of big holes. And the main constraint here, because of that, we get a lot of compaction happening. There's boron toxicity and a lot of unpredictable rainfall, so we can get intermittent drought. In the high rainfall area, there's more than 550 millimeters of rain. The soils are soil and chromosol. And due to the high rainfall, there's a lot of waterlogging constraint, as well as acidic soils and all the toxicity of aluminum that comes with the high pH in this particular area. The pulses that are grown within this region range from field pea, chickpea, lentils, and faba bean. In the drier regions, farmers and agronomists have tend to lean more towards chickpeas as the dominant crop as it grows better in the drier region. Field peas are generally distributed throughout. Lentils dominate the mid-rainfall region because they are able to grow here the best and have the best yields. Whereas faba beans we found were mainly in the high rainfall area as they were able to tolerate those waterlogged conditions better than any of the other pulses, and it grows best under high rainfall. For the purpose of our study, we had selected field pea and lentils because we've got field pea and lentil breeders within our group, and this meant we just had their resources to rely on as well. And using that germplasm, which has been released in Australia since the domestication of field peas and lentils, over here, we tried to characterize the above and below ground characteristics. And our team member, Viri Silva Perez, she identified all the above ground root traits, while above ground pulse traits, while I looked at the below ground stuff. And above ground field pea, you can quite easily see visually that there's quite a bit of difference in the above ground traits. In lentils, it's a bit harder, but she did manage to identify that you have very erect type of varieties such as Nipa that have very narrow angled branches, whereas PBA Bold has very spread out above ground architecture with spread out branches. However, very little is known about the root architecture, and this is mainly because roots are very hard to study. You need to dig, wash, scan to get to the data, whereas the above ground architecture, you can take images, you can visually score them, and it's a more high throughput way of identifying the traits. However, roots are important and we do need to study them. I feel like if you look at only shoot traits and not root traits, you're going to a battle without a shield, just a sword, which doesn't work. You need both. You need to address both to get the best outcomes. And we need to look at roots because roots contribute to above ground plant traits. It is the source of extraction and extracting nutrients and water and supplying this to the plant for yield. And they are also at the forefront of all the constraints. And if we can address the constraints where it occurs in the soil, then we can address our yield gap better. And overall, we just need to look at roots because there's not enough information available on roots. And this is just a little exercise for spot the difference for all the viewers. I did leave a little bit of clue, and to spot the difference, these are the 4 pulses I have been talking about. If you can take a guess, that would be great. If you've worked on pulses, you can probably guess some pulses and roots. If not, I've left a little bit of a cheat over there. So moving from the left side, we've got a pea root system. Then we've got nugget, which is a lentil I talked about earlier on. And then we've got the chickpea. And lastly, on the far right is the field faba bean with a quite profused and big, nice looking root system. And to characterize all these root systems, we look at different traits. And this is just the sphere of all the different traits involved in root system architecture. You can look at the morphology, physiology, and phenology. And I'll be talking a lot on the morphology side of things about the total root length, the total root surface area, and so on. I haven't looked at physiology, but a lot of studies have been done on root physiology, especially in terms of water use efficiency, because that is key to drought tolerance. Phenology has been a little less touched on, but it is emerging more and more in the field of root research. From the earlier desktop study where we looked at what constraints existed within Victoria, the soil types, the weather conditions, we came up with an idiotype that would best work within our agricultural system. There is a popular idiotype that already exists, which was proposed by Jonathan Lynch. This was the steep, deep, and cheap root idiotype, which basically means that if you have moisture available in the subsoil layer or deeper in the soil, then during a terminal drought, the plant can grow deeper roots and access this subsoil moisture, which will help avoid the stress imposed by drought. And to get these deeper roots, you need to have a steep root angle so that all the root growth is directed straight downwards, have a cheaper root form. So because we know that roots take up at least twice the amount of photosynthate than above ground parts do. So you do need a very low investment root growth, but a root growth that does reach the subsoil moisture to avoid drought. And this particular idiotype is very good for a system where there is moisture available, and that is mostly in the northern cropping system, such as in Queensland, further north in Australia, where it has a summer dominant rainfall and dry winters, but we do get large amounts of rainfall before the cropping season starts. So there's enough water available, and they often have long fallows. So that's all good with the cheap, deep, and steep root idiotype in that particular cropping system. However, in Victoria, the cropping system's a bit different. First of all, we've got a compaction layer. Then our summers are quite hot and dry, and the rain that we rely on comes during the cropping season. It is a winter dominant rainfall. And we have a lot of continuous cropping happening, and there's limited subsoil moisture available because of the continuous cropping and the unpredictable rainfall. We just don't have enough subsoil moisture available. And so a deep root system would actually cause harm in this particular cropping system if the plant is investing into growing deep roots and there is no subsoil moisture to tap into. So the idiotype that we've proposed through our study is a more smaller root system with a very wide root angle so that the root can spread more horizontally. This would create like a mat effect on the topsoil so that it can capture all the moisture from those intermittent rainfall events before it is lost due to evaporation. And so the aim of doing the characterization was to find traits within genotypes that can help us build this particular idiotype in lentils. Oh, sorry. Yes, so I've conducted, in the small time that this project has been on board, I've conducted a few experiments. The first one was a clear pot seedling screen. This was mainly to see if there was any significant difference in root angle, because root angle is one of the major factors in the idiotype. And basically, I looked at 36 genotypes. Seedlings were grown for 2 weeks, and then they were imaged in the pot, and the root angle was determined using ImageJape, because the minute you take out your seedling and wash them, you lose the structure of the root, and then it's hard to determine what root angle it actually has. But once the seedlings were washed, they were scanned and analyzed using a Mycorrhizal software. And as it was in a seedling stage, you can characterize the different root parts. So you have the long taproot, the lateral root, and it can identify all the secondary order lateral roots and so on and so forth. Unfortunately, there was no significant difference in the root angle from this particular experiment, but we did find that there was genetic diversity in terms of the root length, root surface area, and root biomass. So that was a good indicator of variability within the germplasm. The next stage was to take it up and look at it at a mature plant level. So we grew them up in these big PVC 1.45 meter pots. We grew them out to maturity, and did the same process. You can see that it was wrapped up in plastic. So we took out the root system. It basically looks like a long sausage roll. The roots were sectioned into 10, 20 centimeter segments. They were washed and scanned in Mycorrhizal once again, and it allowed us to measure all the different root traits of length, surface area, diameter. And from that study, few interesting things came up. So usually we think of roots just being like mostly distributed in the top 10 to 20 centimeter, being the most dominant in that region, and then tapering off, creating a top-heavy approach, so to say. However, I found that the root distribution differed quite a bit in lentils. In fact, there were 5 root forms that I came to see. The first one is a shallow, top-heavy, sorry, 1 second. Yep, a shallow and top-heavy root form, and this particular root form is beneficial for in-season rain and having where there is phosphorus deficiency, having it in the top layer, because phosphorus is mainly contained in the top layer. It's not soluble, so it doesn't move down the soil profile as much. So where you have these particular types of constraint, a top-heavy root distribution would be quite beneficial. The second one identified was a deep and bottom heavy root system. So if your roots were mostly dominant in the bottom section, such as this variety nugget, it would be beneficial where you had subsoil moisture available at depth and to access nitrogen. The third one was a middle heavy root system, and most of the roots are in the 40 to 60 centimeter zone. And this particular root form is beneficial if you are looking towards herbicide tolerance or avoidance, because herbicide does tend to move down the profile after spraying, and it can cause root death, which can result in plant death. So if you have roots which are protected and majority of it is further down, it would be a beneficial trait. The fourth one was a hourglass shape, and this one is like having your feet in 2 boats at the same time, which is the distribution in the top layer and distribution in the bottom layer. So if you do tend to have subsoil moisture, the plant can access it with the distribution in the bottom half at depth. And if there is no soil moisture available, it can maximize with capturing moisture from rainfall by being top heavy as well at the same time. So it's very good for either form of intermittent or terminal drought scenarios. And the last one was a straight root structure where the distribution seems to be pretty even, and this can be used in combination with other traits. So just quickly, a few findings from the 2 studies was that thin roots grew to a greater depth, and there was a lot more biomass investment in thin roots. These were mostly varieties that we initially got from Canada, like Commando, Aston. These varieties had a long maturity period. They put on a lot of biomass above ground as well as below ground, and the roots were deeper and thinner. Also, root angle had an impact on root volume and root length and surface area, which basically indicated a good investment in root growth. There was significant diversity of shoot and root traits across the 36 genotypes. So basically touched on most of those. In addition, we also found that genotypes, which were generally taller, put on a lot more branches, and they had pods at a higher height. This pod height was advantageous because if you don't have pod at height, then you lose a lot of pods when you are harvesting, as the harvester cannot go so close to the ground. So it was a good trait to see in relation to roots as well. And finding of the 5 root forms in general, along with the finding of lentils having thin, long lateral roots in general across the board. And then just quickly moving on to the root measurements in the field. So traditionally, for all my studies, I have to go out in the field, take root calls, and do the core break method, and count individually all of the roots that are present in each of those 10 centimeter segments. This takes 4 people over 5 days to finish 100 plots on estimate. So it's a lot of manual labor, it's a lot of time investment, energy investment, and yeah. So a way to save on this was to use the root imager CI600. If we have them installed, we don't have to take the Cora out every time. We don't have to manually count all the roots out in the field. So it was a lot of time saving from being out in the field. And also it allowed us to take root measurements throughout the season without damaging the plant. As you can see in the picture on the left-hand side, it's a pea plant breeder's plot, we call it. It's a small 5 meter by 2.5 meter plot. And during the season, we go and take biomass cuts. However, it's very hard to take a cora in there and then go and take soil cores to take root measurements, because we'll be damaging all the other healthy plants that are remaining in the plot, because these plants tend to have to lodge sideways, they go off the track, or they get tangled onto the cora itself. So to avoid all of that, we decided to use the root imager. And I started off doing a preliminary trial last year, and we did it in a irrigation trial. So basically, from the 5 root forms that I identified earlier on, we took representative varieties, grew them in a rain-fed condition and irrigated condition. So since this was a preliminary experiment, we treated them separately. So since this was done in 2020, there was a lot of difficulties. First of all, we planted our plants and we did not order things did not come in time. So there was quite a bit of delay. And as you can see, the plants are quite big in the bottom pictures when we started installing. And the lesson learned there was that we need, for at least these crop plants such as pulses, the tubes need to go in straight after sowing, so there's least amount of disturbance to the roots and to the soil structure itself. We also wanted to have longer tubes, so we had them custom made locally, as was our company policy. And these tubes, they were made to the correct length. However, there was a little bit of mistake in the diameter, and I realized how much of a difference 5 millimeters can make, because when you're dealing with soil and taking soil cores, it is such a bulky thing to do. There's so much of soil disturbance. You don't think that 5 millimeters is going to make a big difference, but we could not install the plastic tubes when we took the correct cora size, just because of that 5 millimeters. So this year, when we are redoing the trial properly, we've got all the proper tubes and it went smoothly without any issues. We had extra long tubes so we can make sure that we get the maximum rooting depth for all of these lentils and field peas. The other issues we ran into was that, because as I said earlier on, the soil is a vertisol in this particular region and it's very sticky, especially when it's wet. And we have a screw cora, so it rotates as it goes down to create the hole for tube installation. And as you can see in the top right-hand corner, metal broke as if it was wood. So there was a lot of auger breakdowns, and the lesson learned from there is to have a fabricating engineer just on speed dial, because you'll be going there twice a day to get things fixed, one thing or the other. Other things that we learned was that, I think I've covered everything in this particular one. Oh, also that because the tubes were bigger, the caps fell in. We lost a few tubes due to the cap being blocked in the tube. Moving on to the image processing side of things, I think Dr. Elmi has covered a lot of the things, so I don't have to cover a lot of them, but if there's anything different, I'll just state those. So as you can see on the far left-hand side is a stitched up image of all the 4 windows. With our longer tubes, we can have 5 windows in there, so it goes a bit more in depth. But for this particular season, our roots didn't go that deep. Because it's a screw cora, you can see all the diagonal lines even occurring in the image. One of the lessons learned from here was that we get condensation throughout the season. So once the tubes go in winter, that's when the season starts, all the way to summer, there was condensation happening. Also, there was condensation because it's a cracking clay, and as the soil dried up from winter to summer, the soil went from being stuck onto the tubes to like shrinking away from the tubes, and it created a bit of distance between the scanner and the root. So the roots were a bit harder to see, but we didn't manage to trace up because there's a human person actually tracing the roots. They were able to make those judgment calls and do tracing for them. So that was one of the issues we ran into. The only solution I had for that was at the beginning of the day when we go into the field to remove all the caps and allow a warm air to get into the tube and sort of homogenize the temperature and reduce a bit of the condensation. The other issue I ran into specifically because of having the wrong size, that 5 millimeter difference, was that little bit extra diameter created a little bit extra distance between the inner surface of the tube and the scanner, and the light was able to bounce back and reflect and created that white streak down the center of the tube. We didn't manage to work around it, but with this year's proper trial, we don't have that issue. We just used better tubes to solve that particular issue. Another lesson learned that I forgot to mention was that for this year, we did find that there was a lot of root material present in the soil from the fallow before or from the stubble that was left onto the soil, because as you make the holes down the soil, it goes in a screw motion, and sometimes you have plant matter go down and stick onto the walls, and later in the season you don't know what was root from your experiment and what was pre-existing stubble or root. So we've gone in straight after installing the tubes and taken an initial image, so that when we take the next image and the following image, we know what was pre-existing and what is actually ours. Yep. And this is, though it was just a preliminary trial, I did want to see what differences I could pick out from the rain-fed and the irrigated system. And there was quite a bit. So generally the rain-fed ones had roots down to a greater depth, and there was less variation in this particular root trait measure with the CI 600. With total root length, it really depends where your tubes are being placed. So because we placed it in between rows, there was very uneven distribution. This year, we've made the point of installing it directly underneath the plant. I saw a question earlier on why is it installed at 45 degrees. Basically, if you install it at 90 degrees, you will only get lateral roots. So if that's something you're interested in, just to see vertical distribution of lateral root, that's when you'd install it at 90 degrees. At 45 degrees, it allows a perfect snapshot of what the roots are growing across the tube lengthwise. And because it's an angle, you get a length and depth perception of what root distribution is for that particular treatment. So yes, installing it directly under the plants for these small crops where you take them out at the end of the cropping season allows you to get maximum root capture. A better thing would be to install more than one tube so that you can increase your sampling size and reduce the sampling error and get a better idea of what the root distribution is. The other trait I looked at was root angle. No significant difference with the root angles, so to say, with the irrigation and rain-fed. Once again, this could be because the tubes weren't installed directly underneath the plant. So I'm hoping to see a bit more correlation with the root angle and depth in irrigation this year with the proper installations. Average root diameter. This basically showed that with most of the varieties, the rain-fed ones had thicker root diameter, the irrigated ones had thicker root diameter, whereas only Digger and Northfield, the two varieties, had thicker root diameter in the rain-fed scenario. Looking at the correlation, so while exact measurements can be subjective, I think trends using the CI 600 is a good trait to see because trends don't seem to fluctuate as much. And it was good to see that certain things like root count and total root length remain pretty consistent in, the top one is, I forgot a label. The top one is supposed to be irrigated correlation, and the bottom graph is rain-fed correlation. And in both situations, root count and total root length had a strong correlation. This is something I haven't picked up when doing manual root counting, but with the CIC, with the Rootsnap software, it gives you root count alongside with root length. So looking at the data that way is much easier, and it allows you to pick out things that you wouldn't pick out otherwise. The next interesting thing that I saw was that in a rain-fed situation, root count and total root length had a negative correlation with the average root diameter, indicating that in rain-fed conditions, roots were generally thinner and longer, whereas in irrigated condition, roots were shorter and fatter. Also, branching angle and branch count were positively correlated to root count and root length. Other correlations was of root volume to area and branching angle with branch count. Other noteworthy mentions that didn't make the significant mark, but may differ depending on the amount of water, because where water is limited, root traits tend to govern yield and above ground characteristics a bit more compared to when there is an excess of water. So in the rain-fed trial, we saw that minimum depth had a negative correlation with total root volume and area, but where water was ample, this particular correlation did not exist. With ample water, depth had a potential correlation with total root area and branch count. Also, average root diameter tend to be negatively correlated with root count and total root length in a situation where water was not limiting. Okay. So in conclusion, when looking at the mini rhizotron data with the root imager and comparing it to washed out roots from the coring, there is of course a difference, because the coring system, we take a lot more cores, it's washed, whereas with the rhizotron, we installed only one, so it only had one sampling compared to the 4. In addition, that the roots are being traced, and it introduces a human error. So if you are adapting the software, one of my recommendations would be whoever is tracing the roots to be very consistent, so it allows you to have more confidence in the data that you have, that they understand the software properly as well. And but in saying that, despite the difference, if you are looking at general trends, the general trends were comparable of what I found with washing and what I found with the image analysis from the root imager. In comparison of root images showed a variation from genetic influence, so we saw that root diameter, root length, root area, volume, and even branching angle seem to differ between the 5 varieties that were tested in the different treatments. And overall, irrigated plants just had greater root area, which was due to root diameter. And these irrigated plants dominated the first 2 windows of the shallow zone with a lot more roots, thicker roots, whereas the rain-fed ones had thinner roots in the top 2 windows, and they had root distributed in the 3rd and 4th window as well, showing greater rooting depths. And so overall, I'm just hoping for a better trial in 2021 compared to 2020. And we'll see how that goes. And this is just an acknowledgement of my team, my immediate small team, and then the big pulse breeding team at Agriculture Victoria. Thank you.
All right, thank you so much, Dr. Rao. That was incredible. Also would like to acknowledge the fact that it is 2 o'clock in the morning where you are right now currently. And so we really, really appreciate you being on this webinar. And I hope everyone else appreciates that as well. Let me go ahead and finish up. We'll wrap up the presentation here, and then we'll go right into the question and answer session. So we heard a lot from both of our researchers about the specific applications of their all the different projects that they're working on with the root scanners. And I think the one biggest takeaway, one big takeaway that we can take away from this is that there is no general solution to installing your tubes. There's no general practice. The only practice that we can recommend, generally speaking, if you want to get a whole picture of your roots, is to install the tube at a 45-degree angle. But otherwise, as you can see, there's so many variables: soil conditions, climate, your microclimate, the type of plant that you're studying, and there's so much variability there that there is no just one solution fits all kind of situation. So you do need to have and as Dr. Lotze said, and also Dr. Rao both mentioned, you need to have a good understanding of your specific crops or plants that you're studying, your soil conditions, your climate, and all of that, and to kind of get an idea of how you think you should design your tube installation, and all that. So I think that another great idea, if you are interested in this and you aren't sure about tube installation or where to install the tubes, how to install the tubes, how long of a tube, whether you should install it vertically or at a 45-degree angle, all those questions, I highly recommend you find other researchers that have done things similar, or at least somewhat similar to what you're doing, and try to see if maybe you can get ideas of how, learn from their experiences, just like we learned today about, especially when it comes to trying to auger in clay soils. I think both Dr. Lotze and Dr. Rao could give you a lot of feedback about how to best go about doing that. It sounds like it's a little bit of more of a hassle than you might think. So, but again, thank you everyone for joining us. We will go into a Q&A session here. If you do want to learn more about the instruments, or you want to learn more about what our company is or what we do at CID Bioscience, feel free to get connected to us. You can email us, sales@cid-inc.com. You can always visit our website. That's where we continuously update anything that we're working on. Follow us on Twitter or on Facebook or on LinkedIn. You can also find us. And we do have a facility. Due to COVID travel restraints, we aren't necessarily recommending you come visit us, but if you already happen to find yourself in this area, please reach out to us if you want to come visit, and we'll be happy to try to accommodate that. And lastly, if you do want a quote for an instrument, or if you want pricing information, please click the tiny URL link that Scott will be posting in the chat. And also, if you would like to request a consultation, you can do that as well by clicking the Outlook bookings calendar link right there. That'll be a consultation with either myself or our other application scientist, Eric Munoz. And we'll be more than happy to answer any questions you have about your specific application or anything like that. Also, if you would like a quote, you can also always email our sales team, sales@cid-inc.com, or give us a call here at our facility. So let's go ahead and open this Q&A box. And we'll start from the very first question that was asked, and we'll just kind of work our way down. If there's any questions that are specifically addressed to Dr. Lotze or Dr. Rao, then I will be sure to make sure that they know that, and they can jump in whenever the time is to answer those questions. The first question is, to what extent the root imager can detect and scan the root architecture? I'm not sure the specifics of what you're asking. So the scanner can only essentially scan or image roots that are within a couple millimeters of that tube or touching the tube that's installed. So it's not like a penetrative image like an X-ray or anything like that that gets an entire image of the roots. It's just wherever that tube is installed, anything that's within a couple millimeters of that tube can be imaged. The next question is, can the CI600 and 602 differentiate the primary, secondary, and tertiary roots and root hairs in volume, weight, and size? So the CI600 itself, the instrument itself, only gives you an image. The analysis software that you use may or may not be able to do this. The Rootsnap software does not necessarily give you the information of this is after you trace them, doesn't say this one is primary, this one is secondary, this one is tertiary. Root hairs in general are going to be very difficult to see unless you use the highest resolution. And still in that case, you still might not be able to actually individually trace those out because they are so fine. That might be more of an issue. Weight is not something that any of them necessarily, any software that I know of can calculate, but you do get volume and size parameters, and you could estimate the weight based off of those. But yeah, that's as far as the analysis software goes, that's the kind of the capabilities of that. The next question is from George Richardson, and it's, can you use these scanners to study mycorrhizal images, growth, and relationships? Yes, you absolutely can. For the mycorrhizal stuff, as Dr. Lotze mentioned, you likely are going to have to experiment to see what's the lowest possible resolution you can go down to, while still maintaining the resolution you need to be able to see these any sort of fungal or mycorrhizal relationships. And keep in mind that the file size increases as you increase your resolution, as well as the scan time. And so it's that kind of balancing act of what's the minimum necessary resolution I need for this application. So it really also depends on your plant and the size of your roots and all that as well. The next question was a follow-up that, can you use these scanners to study native plants in the wild, and what kind of depth can you go? Yes, we have a lot of people using these in applications looking at transitions from forests to savannas, as well as looking at tundra grasslands. And so you can use these, you can install these, just keep in anywhere you want to, these tubes, but keep in mind, and Dr. Rao also kind of touched on this, is that there is, when you do install an existing plant system, you are going to disturb those roots. And so you need to do your best to try to minimize that disturbance. And also you got to keep in mind that you got to wait till after that stress response kind of goes away and the roots kind of reacclimate to their situation before you can start getting meaningful results that actually correlate to whatever you're studying, as opposed to something that's in response to the stress of being disturbed. But yes, they are used. What kind of depth can you go to? So the tubes come standard at about 1 meter long. You could get longer tubes potentially. We don't really recommend going much deeper than like 2 meters is kind of the maximum we would recommend, and that's even probably pushing it a little bit, mostly because of the difficulty involved in how many times you're gonna have to scan. Because the scan window is only about a foot long, which in centimeters, what is that? 25 centimeters or something like that long. Then you'll have to do multiple scans, and when you have 2 meters of tube, that's going to be pretty time consuming to have to do for one single system. The next question from Sylvia Mitchell, are the roots confined to grow in the long tubes once they start growing into it, and how long are the tubes, and are they placed vertically, horizontally, or at an angle? I think we've already addressed, yeah.
Shawang had a comment.
Oh, yeah. Sorry.
I was looking for the raise hand icon on this one. Anyway, just wanted to mention with the how deep you want to go, another consideration apart from the 25 centimeters, that the time trade-off, because each scan, I use a 300 dpi that works good enough for me to get the image quality I want, and that has a, if I remember correctly, 60 or 90 second time to do the scan. And in such a situation, I can only finish within the day. If I go nonstop, I don't look at my phone, I just continuously keep going. I can finish 3 rows? Yeah, 3 rows, approximately 36 plots, sorry. Yeah, so it does take quite a bit of time. It is only just taking an image, which you think is a very quick thing, but it does take a bit of time as well.
Yes, thank you for that insight. Dr. Lotze, you have something as well?
Yes. Unfortunately, I have to leave. So
Okay.
please accept my apologies. But please, if there are any questions, you can contact me, and I will make sure that I answer them in person.
Absolutely.
So if that's fine with you.
Yeah, thank you so much for joining us. And yeah, well, I really appreciate it.
Thank you very much for the invitation, and good night and good morning to everybody. Thank you.
Thank
Bye.
you. All right. So, and I think actually now that we're at the point where probably most of these questions are for Dr. Rao or myself. So the next question was, so are the roots confined to grow in the long tubes once they start growing into it? So actually this was something that Dr. Lotze encountered, is that your roots aren't going to be, they're going to grow around the tube, and they're going to keep growing at a farther depth if you're looking at certain root systems that grow into, deeper depths. So what she encountered was that, she was seeing her root systems fine for a while, and then at a certain point they started to go so deep that she was no longer seeing them, but they were continuing to grow. So having that knowledge, you need to then decide whether you want to have multiple tube installations to accommodate the roots growing to further depths, and being able to see them at different stages of root growth, or exactly whatever the specific results that you're aiming to get are. You have to consider that when you're thinking about installing these tubes. And as Dr. Rao will attest, there are lots of things you probably didn't think about when you go to install them, and then it's something like, I guess that is something we got to put into consideration. So it is kind of a trial and error thing for most people, and I will say that off the bat. It can be a little bit of trial and error to figure out exactly for your plant systems, especially if it hasn't been done before and you have nothing reference to go off of. Then it is going to be a little bit of a trial and error situation. The next question is, were the lateral roots segmented from these images with the standard software? So with the Rootsnap software, it's not going to do any sort of differentiation necessarily. It's not going to tell you, like, as the same with the primary, secondary, tertiary thing, it's not going to tell you those things. You'll have to just keep that you'll have to do that manually by human eye. You're not going to be able to have the software itself doesn't have that built into the calculations, I should say. There might be other softwares out there. So as I mentioned, our images can be used with any software out there. So if there is a open source software or Winrizo or whatever other software you might want to use to try to get other insights, then you can absolutely use that software if you would like to. Dr. Rao, I think I saw you.
I'm just going to do this to get your attention for
now.
Yeah. The thing is, like, at least with all my images, you sort of don't get like a whole lateral root coming about and see it branching off. For some, yes, for not all of them. So it may even be difficult to do it by eye in that situation. Mm-hmm. Also, on a side note, I saw a lot of the conversation about, can this be automized to some extent, and I attended a recent root conference, and they showed something called the Root Painter, and this is a machine learning software. So you train the software with 100 or so images, and then it is able to distinguish with the forthcoming images. So you won't have to go and manually do stuff, but I don't know how well it works. This is really in its early stages. I myself am trying to just learn how to use it. So it's something if you guys are interested in.
I did see also, and actually I'm going to really quick skip down because I did see that from the same person, and I saw it in the chat as well, a discussion about the machine learning. Wouldn't it be possible to train a deep learning network to segment roots irrespective of background or root color? As someone who personally, with other products that we work with here, that I work with machine learning and things of that nature, especially with training predictive models, it might seem like something that's very simple to do. It is, but due to the variety and all the variation and all the variables that are present, especially with roots and different types of plants, different root morphologies, soil conditions, soil colors, root colors. It may be possible to do it for a specific, I mean, it's going to be easier to do it for a specific root system. If you wanted to say, this is the only root system I'm studying, but for someone like Dr. Lotze or Dr. Rao, who's looking at all these different types of morphologies and all these different phenotypes of these root systems, it's a lot harder to incorporate all that variability. It takes a lot more work to incorporate all of that variability into a deep learning or machine learning system that will then be able to accurately predict or do things like accurately distinguish background from the root system, or accurately automatically trace and measure those roots. So that's why it is 2021 and we still don't quite have something like that works for any application, because it's that variability alone is the hardest thing. And I think that what the system that Dr. Rao just mentioned is nice, is that it is trying to tailor it specific to your plans, which makes it a lot easier. So you'll be able to train with 100 images of your specific kind of system that you're working with, and then from then on it will work fine for that system. But if you went to go say, okay, well then next year I'm actually also incorporating a whole new crop to the study, and you try to use that same algorithm, that same learning network to identify those roots and do all the measurements automatically for you, it probably won't work as accurately because it's a totally different root structure and everything. So that is something to keep in mind. We will get there. I think that I'm hoping it'll be soon, because this is the most commonly asked for feature of studying roots, because ever since we transitioned from destructively studying them to non-destructively studying them, now it's how do I not spend my entire time tracing roots manually is like the number one request for people. So that will be something that will be available here soon. I think that, like what Dr. Rao said, we're really getting very close to having something like that. But getting back on track here, a couple more questions about, there's a question here about how many tubes do you install per treatment? Again, there is no right or wrong answer to this. It is very application specific. It is specific to your crops, how deep your root systems go. You know, sometimes you can get away with one bigger long tube. Sometimes you need to, I've seen situations in like a breeding situation where they actually developed a custom planter box that had about 12 different tubes installed horizontally so they could get a complete view of everything that was happening within that box. And so it's really, and people, I've seen them used completely vertically in aquaponics situations. So it's really, there's no right answer. I can't give you an answer I mean, you look at previous research that's been done, maybe in your field with your specific crops or your plants that you're studying, you might be able to get a better idea of how many tubes might be required. The next question is another, how many tubes used for one tree crop for good results measurements? You can get away with just one. You can also install multiple. It's really highly specific and application dependent. So there's no, I can't give you a one size fits all solution for that, unfortunately. Where is the ideal point to place the instrument at the plant base, especially on trees? So again, there is no right answer for this. It really depends on the type of tree that you're measuring and the roots, type of root system, and where do you think the roots are going to be, where are you going to get the most benefit and most root cover of that tube based on the install. I can't give you, unfortunately, I cannot give you a direct answer to that. The next question I feel like is, this one's actually for you, Dr. Rao. I think it was in regard to the when you identified the different root zones, the roots that went into different, your ones that were thicker at the top versus the bottom. So this person was asking, nutrients effect in the root zones means which zone volume is high for good nutrients fertilizer. So I think they're asking which zone is the ideal zone in the situation that you were showing, where the roots are going to get the best nutrient absorption and fertilizer effects from fertilizer.
It really depends on your fertilizer. If you're applying something which is more phosphorus, for something which is more phosphorus deficient, if you're applying phosphorus, then you want a top heavy root system, because phosphorus doesn't travel down. If majority of your roots are occurring at a depth, then you may be putting phosphorus and it's layering on top, but your roots are not getting any access to it. If you're talking in terms of nitrogen for pulses, you don't really need nitrogen, they make their own, but in other cereal crops and so you need deeper roots or a bit more root distribution at depth, because nitrogen does tend to mobilize with water down the soil profile. So I guess it depends on the fertilizer that you're using.
All right, thank you for that. And then the next one is actually somebody specifically addressing you, asking, they have several, from Ricardo, I have several questions about the details of the protocol that Dr. Rao applied to measure the root angle. I would like to know if I may contact or write Dr. Rao for her valuable advice and guidance in this matter.
Yes, anytime. I shall leave my email address somewhere.
Yeah, we can.
If you're okay, I can just type it into the comments. Is that okay with you?
Yes, that's absolutely fine. Thank you.
Okay. Yeah, you bet.
Thank you, Dr. Rao, for that. And then the next question from Kwame is, how do you determine the number of tubes used, especially when doing plant breeding trials to often include many genotypes? As I said, so, and yeah, there is no right answer to this. I've seen people use anywhere from 1 to 20. So it's really dependent on how you want to establish your specific experiment.
Also dependent on funding.
Yes, oh, that's a good one. Yes, that's a good point.
For my small trial, I had a total of 50 tubes distributed across the rain-fed and irrigation, and we do have quite a bit of funding. So, yeah, there's the funding does tend to be a limiting factor in things, apart from all the sciences.
100%. Yeah, that is something to consider. If you can't afford, 600 tubes, which most people probably can't, then you want to be more conservative. Like, you need to be as conservative as possible. And so that really, it really depends on, and then getting strategic with the placement of the tube itself. If you're trying to, if you are limited by the number of tubes, and that was when the placement becomes really important.
Yeah. Also, because there was a mention of breeding trial having a lot of genotypes. Yes, so our breeders go through thousands of crosses, and you can't install that many tubes and invest that much energy. So our approach was to first do a screening, either do a seedling screen or something, to get the genotypes that would most be targeted towards your goal if you're looking for idiotypes or tolerance, and then using that downsized subpopulation to do root imaging and looking at it in depth in the field.
That's a very good point. Yeah, I think that's a very good strategy to, especially as you mentioned, thousands upon thousands of genotypes potentially. There's no one, I don't know anyone that has the money for thousands of tubes. So that is a good point. So the next question is also for you, actually, Dr. Rao, is the root length would be positively correlated with drought tolerance? Question. So yes. I guess, yeah.
It depends on your type of drought. For me, when I initially started on this journey, I was also like, drought is one thing, water stress. But actually, there's different types of drought. You can have terminal drought where there's no rain, but you'd have water available at depth, in which situation having root length directed towards rooting depth. So root length should go in hand with rooting depth to reach that moisture. Then yes, it is a drought avoidance trait, which helps the plant survive such a drought situation. However, if it's a intermittent drought where you don't have subsoil moisture, then depth is no, it's not a good thing. You want all that root length invested in the top layer so that it can capture any amount of rain that comes through and it should be paired with an above ground architecture, which is spread and a thick, dense canopy to prevent any evaporation from occurring. So you just want to capture any bit of rain that comes in with a lot of root mass in closer to the topsoil layer.
Awesome, thank you for that. The next question is, what could be considered as the maximum number of tubes that could be installed for practical research that is manageable? Honestly, for most applications, you can easily just use a single tube, and even in some cases, a single tube for multiple plants. So I think that you don't need to, there are ways to get around using multiple tubes. And as we mentioned, that is there is the financial and time limitations of having that many tubes installed. If you are really concerned about it, and like in Dr. Lotze's situation where the roots grew past the initial tube that they had, you could put a second tube. But I don't think there's many situations. As I mentioned, there are some people out there that have created boxes that have, literally 12 different tubes going horizontally through them that allows them to see, a really total image of everything that's happening inside that planter box. But that's a very one-off kind of situation. I feel like that's not a very common situation to see. So more commonly, you see one tube per plant, and even one tube for multiple plants potentially. The next question, are there any videos or documents to demonstrate how to use the Rootsnap program? We do have a manual available on our website, so even if you don't own it yet, you just want to know more about it, there is a manual on our website for Rootsnap. I don't actually know if we have a video or not. That is a tutorial walkthrough video for Rootsnap, but we are working on making improvements to Rootsnap. So with those, we will likely have an updated tutorial video along with that. The next question, the condensation droplets are very white, so using a low pass filter can remove them, not affecting the roots because they are not as white. So some good insight from Leonard, that there is ways to also get around condensation. If you don't have the time to go and condition your tubes like Dr. Rao did with taking out the caps and waiting, then you also can do things like actually go and either physically try to use a towel and remove them, or use a low pass filter to help remove that, or any other kinds of filters that are available. Because it is just imaging technology. You can use filters, and sometimes that might help with the differentiation issue as well. The next question is, it can also be used in fruit trees to measure roots in mango trees. It's been used in a lot of orchard applications, as well as it's been used in citrus. It's been used in apples, as you've seen. It also has been used in other forestry applications, so it most certainly can be used in mango trees. Can you use these scanners to measure teff root crops, which is the grass family's crop? I personally don't have a lot of experience or know many. I know that we have people looking at tundra grasslands, and they've had success using it. I think with grass, the hardest thing, and Dr. Rao, I think you mentioned this as well, and maybe Dr. Lotze as well, but is differentiating roots is not with just images, is something that's difficult to do unless you have a very trained eye. So if you have a mixed system where it's not just your specific crop that you're looking at, there is the potential, if you have, if you have weeds or other, other plants that are there, you might be getting a system where you might be overestimating your roots because you're looking at other species that aren't your target crop. So that is something to keep in mind. You can use it for grass assessment. We have people, like I said, in the tundra using it for that specific application actually. Like I said, I think the mixed system thing is something to be aware of. And then the next question is, can you use it to study the correlation between the soil-borne diseases, wilt, and lentil genotypes response? So I think this is geared towards you, Dr. Rao.
Yeah, I guess so. If you can see it, you can measure it.
Yeah. It is a visual technology. It's an image. So yes, as Dr. Rao mentioned, if you can see it, that's when you know you can measure it. The next is a statement. Thanks to Dr. Rao and Dr. Lotze, and to me, very interesting. So a good thank you from our viewers. And funding is the key. Yes, it is. So, the next question is it applicable for leafy crops? It's applicable for anything that has roots. You can use it for anything that has roots. And then the next question is root to shoot correlation. And Dr. Rao has looked at this. Dr. Lotze was looking at this. Everyone that looks at roots also understands the idea of a holistic approach to their research, where they also should be looking at what's happening. It's a whole plant picture, above ground and below ground. So while they might not be specifically looking at, they might have partners that are also, you know, researching the shoots, but correlating those two always needs to be done in order to get that whole holistic plant image and knowing what's happening with the plant. And that is our last question. So thank you everyone for giving us these questions, and thank you so much, Dr. Rao. It's so late there. I really appreciate you being on this webinar and sticking around for the question and answer session. Thank you, everyone. As I mentioned, if you are interested in pricing, please click that tiny URL to go to our quote request page. If you want a consultation, if you still aren't sure whether the root scanner can be applied to your specific research, then please feel free to book a consultation. Either myself or our other application scientists will help determine whether or not our solutions are correct for you. And again, thank you to Dr. Lotze, and thank you, Dr. Rao, so much. Your research, we're really looking forward to seeing this year. Hopefully, good luck this year with the actual imaging, and yeah, looking forward to seeing what kind of progress you guys make. So thanks again for joining us, everyone, and have a wonderful day.